Magandang gabi mga omin na mahal ko. Welcome to a new series that I'm gonna do probably like sometimes, occasionally. So this is called Corn Academy. Okay, so we're gonna be talking about biochemistry of amino acids. Just a brief intro. Okay, what exactly are amino acids? They're protein building blocks or monomers. Of course, if you start building them up, they start becoming dimers and polymers. They're composed of this guy, the acidic group, called the carboxyl group, this guy, the amine group, and the side chain, called the R group. They change, like the side chains, the R groups, they change all the time. They're also Zwitter ions, so when they become something called ionized, you have a negative charge and a positive charge. And yeah, a lot of this is like assuming that you know organic chemistry already. So yeah, a lot of biochemistry is organic chemistry, just combine both bio and chemistry. Yeah, also amino acids are called residues. Okay, amino acids are also chiral, except for one of them. So 19 of the 20 amino acids are chiral. They're non-superimposable image. So imagine your left hand and your right hand. You can't like cross them to each other. Yes, this is Jojo Zahando. So yeah, you have your left hand, right hand. You can't really cross them together. You can't superimpose them, and that's what makes it chiral. And it's important in a lot of biological bases and stuff. A lot of, like, all of the amino acids in nature are L amino acids. So amino acids come into two forms, the L and D amino acids, the levorotatory and dextrorotatory isomers. So we use this corn rule. It's kind of a coincidence I kind of call this Corn Academy, and this is the corn rule. So you go clockwise, and you have carboxyl, R, and your amine. So you go corn, CORN. Also, this is called the alpha. I forgot to talk about this. This is called the alpha carbon. The alpha carbon, it's called the alpha carbon because you have the carboxyl group over here with the O, and anything that's right next to it that is a carbon is called the alpha carbon. And yeah, oh snap, it's time to memorize all 20 amino acids. There's like so many, but one like good thing to like note with this is like all of them are, well, a lot of them are similar to each other, so you can start grouping them up. So like you have 5 here, 2 here, 3, well, 2, 3, 3 here, hydroxyls 3. Yeah, you can start grouping them up. I based this on 2 of my notes. So my cell biology class also talks about amino acids, and it kind of says like different things compared to my biochemistry class. But yeah, let's go through them. So glycine and aliphatic residues. So these guys are kind of sort of non-reactive, mainly because you have hydrophobic groups like methyl, isopropyl, whatever this is. Glycine is the only achiral amino acid. It's really small. And then you go alanine, it's a methyl group. Valine, a good way to remember that is there's a V right here. Leucine is just a longer version of valine. And then isoleucine, you just start the branch off somewhere else. Yeah, I call them say-so amino acids because they're pretty chill. You know, they don't do much. They're very unreactive. Well, kind of unreactive. Also drink water. And they're hydrophobic. Yeah, next are sulfur residues. So methionine is like one of the only ones. Well, compared to cysteine, methionine is more unreactive than cysteine. The reason why is because this SH group right here, the sulfur, is kind of like an oxygen. Well, if you see it on a periodic table, you have oxygen and then sulfur. Sulfur is like in the same group as oxygen, so it kind of acts like a hydroxide group. So the thioether is sort of unreactive. Also, methionine is like the beginning of any translation. The start codon, if you remember that from your basic bio class. Next is cysteine. These guys can make disulfide bridges, so like curly hair, that's what causes that. Also cataracts. It's mainly because of like the redox reactions that happen between two cysteines, which form cysteine. Yeah, and also other things like bladder, kidney stones. Yeah, and next, proline. Proline is pretty whack. So, proline has a cyclic structure. It's the only amino acid that wraps around itself. It wraps around the amine group. Right here, it becomes a secondary amine. So, this guy, other than like everyone else, this guy is the only one that can have cis and trans that are more so present or like extended compact. So you can find them in turns. You can find proline usually in turns. Also glycine. Glycine is really small. Post-translational modification, hydroxylation with vitamin C. So, other thing that I kind of want to talk about is hydroxylation and vitamin C is scurvy. The problem with scurvy. The problem with that is your blood vessels get thinner or like get weaker as a result of collagen not being able to form. So you really need vitamin C so they can get hydroxylated. It's important for like post-transitional modification. And yeah, I put this here mainly because proline is important for like turnarounds, you know? Turn around bright eyes. Turn around bright eyes. Turn around. Okay. Enough of that. Aromatic residues. So I put these guys because they have the benzene ring. Tryptophan is really thick. You can see a UV absorbance at around 280 nanometers. These two guys are fairly unreactive. And then tyrosine, because of this OH group right here, it's kind of grouped like differently. It kind of just overlaps. The tyrosine can react with iodine to form thyroid hormones. Yeah, iodination. It's called iodination. It's called electrophilic substitution, so like it usually can react right here-ish. And yeah, tryptophan has a indole group. There's also aromatic. It's really thick. You don't see it in turns because of it being thick. And all, she thick fam, extra thick. Next, hydroxyl residues. These guys are the CoOrdinaries. I don't know why I call them CoOrdinaries. Wait, why did I call them? Wait, it doesn't make sense. Wait, I might want to scratch this off. They're not CoOrdinaries. They're actually pretty reactive. They have a relatively high pKa, if you remember pH from your Gen Chem. pKa, high pH, usually more on the basic side. So yeah, tyrosine is also a part of them because of the OH group. Then you have serine, and then threonine with another methyl group in the same group as it is. And also, these guys are the only ones that can do that can be modified with phosphorylation, so that's important in biology. Glycosylation, so glycosylation is adding sugars, so that's important in making stuff like the blood groups, the blood AB, ABO, AB, 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 and O blood types. Next are amides. So these guys are kinda say-so, but kinda also reactive, but not very reactive. Of course, you can do glycosylation. I kinda talked about that earlier. It's mainly because of this thing, this group right here, the CONH2. That group is really stable. So you have asparagine and glutamine. Glutamine is just a longer version of asparagine. You have one more carbon. That's it. Okay, and then negative acidic residues. So if you, wait, let's go back. If you modify this to become a carboxylic acid or a carboxylate salt, or not really salt, carboxylate ion, you can form glutamate or glutamic acid and aspartate and aspartic acid. So these guys are the alter, the alter version of the amide residues. So these guys are reactive, low pKa. So you, of course, they're acidic. They're negative in their ionized form. Glutamate is also a neurotransmitter. If you know that from psychology, MSG and umami, the taste of umami, it's really addictive. Aspartate is just a short version of glutamate. They're in love with lysine and arginine. I'm gonna talk about that later because they're negative residues. And for glutamate, a carboxylation, which requires vitamin K. Kind of forgot what this does. And yeah. So you just carboxylate the glutamate. Okay. So these guys are positive basic residues. So histidine is kind of weird. We're not going to talk about that yet. But these guys are more in the definition of the positive basic residues. You usually see the NH parts of it. Arginine is just weird. And I believe there should be a plus here. Yeah, you can turn this into an NH3 and then you make them positive. So yeah, these are really long. You have 1, 2, 3, 4 carbons, then 1, 2, 3, then another one right here. They have relatively high pKa, so they're basic. They're in love with aspartate and glutamate, the one I talked about earlier, because opposites attract, as you remember from physics. Arginine is also in love with tryptophan for some reason. Oh wait, yeah, I remember that. That's like parallel and perpendicular attractions. So one of these groups is attracted to the aromatic ring of tryptophan. And yeah, mainly because of the cation pi bond. And also, these are important with vitamin C. Also with like collagen and stuff, scurvy. And yeah, this guy is pretty kimochi waroi. Like he's creepy. He's not supposed to be here. Yeah. So histidine is pretty weird. He has a imidazole group side chain. It's useful in nickel and affinity chromatography. It's used as an acid and as a base, so it's kind of not. It doesn't really match like the definition here, but I just put him here just because. Why not? And yeah, it has a pretty relatively low pKa compared to these guys. So this guy is like 6 to 7 pKa. So yeah, next. Okay, we're done talking about the amino acids themselves. Now we're gonna talk about protein primary structure. So the primary structure is just your long chain of amino acids. It's important because you have peptide bonds between them. Peptide bonds are consisted of amide groups. So amide groups look like this: COO and H, and I kind of talked about it earlier. They have a resonance, stable, pretty resonantly stable. They have a dipole. So this guy donates an electron. This guy becomes a double bond. This guy becomes positive. This guy becomes a single bond. This guy becomes negative. And yeah, since we use a Korn rule, we usually read it from C to N terminus. So the C terminus is like the end. Terminus means end. So C to N. Yeah, so C to N. It's very cheap to form a peptide bond too. Energetically cheap. So it's kind of favored, or more so favored compared to other bonds. Yeah, let's move on. Secondary structures. So we have alpha helix and beta pleated sheets. So you usually see this in alpha helix. You have the O being negative and the N being positive. And it goes from the first CO to the fourth NH. Yeah. And then you have parallel and anti-parallel amino acids. So these guys point C to N like that, then C to N like that. Anti-parallel is more stable than parallel, mainly because these guys have 180 degrees of hydrogen bonds. So yeah, I forgot to talk about this, but these guys are formed through hydrogen bonding. And let's move on to the tertiary and the quaternary structure. So protein domains, so just a bunch of polypeptide chains, and they fold independently, they're stable. And yeah, that forms coordinary structure. Sometimes, like, proteins need a bunch of domains to be functional. So you have homomers and heteromers. So they contain different subunits and identical subunits and different subunits. This guy is the structure for hemoglobin. So as you can see, you have this guy being identical, then these guys being identical. So this would be a hetero tetramer, if I recall that correctly. And yeah, that's that's basically all I kind of want to talk about. And yeah, thank y'all for watching. Please like and subscribe. And yeah, I might make another Corn Academy video sometimes if y'all really like it. And yeah, see y'all in the next episode or next video. Otsumium.